Okay, so we're talking about 2.7 DNA replication, and we're going to stick with just replication today. Before we can get too far in, I want to do a quick overview and talk about central dogma. So when we talk about central dogma in theology, we're talking about a central theme or a central tenet of a particular religion. Now, in biology, clearly that's not what we're talking about. However, we are talking about a central theme that carries through the theory of how genetic information is transmitted and expressed. So, biological central dogma really summarizes or encapsulates the idea of how information in DNA influences the traits of an organism. So, we're going from the molecular DNA in your cells to the entire organism, the traits that you see: height, skin color, hair color, eyes, all the physical traits that you see. And not just physical, also we know emotional and behavioral as well is predicated, at least in part, on your genetic makeup. So central dogma is really, again, how we get from those A's and G's and T's and C's to the thing that you see in front of you, the actual organism. And this is true for all living things on our planet. The first part of dogma talks about DNA synthesis. Now, this happens during the synthesis stage in the cell cycle. You might have to reach all the way back to freshman year to think about the cell cycle for a minute. So we have G1, where the cell is growing and doing its thing. We have S, when DNA replicates. We have G2, when it gets ready for mitosis. And then we have mitosis, and the process starts over again. So we're talking about the S stage. Now, after DNA replication in a cell goes through mitosis, and it's going to start doing whatever its job is. So say you're a pancreas cell and your job is to produce insulin. You're going to start producing your insulin. But in order to do that, you have to read the information that's in DNA and you have to transfer it into mRNA. And that's because one of the major differences between prokaryotes and eukaryotes is that eukaryotes, we have our DNA in a nucleus and our DNA stays there. Now, the organelle that creates proteins and actually builds them is the ribosome. And there are not ribosomes inside the nucleus. So somehow you have to get that information out. So the process of transcription is copying the information in DNA into mRNA, which can leave the nucleus, right, and bring it to the ribosome. Then the final step is at the ribosome is going to take the information encoded in the RNA and use it to put amino acids together in a particular order to create a protein. So I know that's a lot, and we're just focusing on this first step today. We're just talking about DNA synthesis. This is the rest of 2.7. So understanding number one, the replication of DNA is semi-conservative and depends on complementary base pairing. And you can also see it over here. So, and I kind of touched on this just a minute ago, DNA replication, in order for cells to reproduce and pass on their genetic material, they have to make a copy of their genome during the S phase of the cell cycle. So if we think back to freshman year and we think about mitosis, we think about chromosomes duplicating, right? Prophase, metaphase, anaphase, and telophase, where they split, so that each cell has an identical copy of the DNA that was in the original parent cell. And this happens during S phase. So here we have a cell. It's going to go through G1. It's going to grow, do its thing. S phase, it's going to copy its DNA, right? G2, it's going to start duplicating its organelles and getting ready to split. And then we go through the process of prophase, metaphase, anaphase, telophase. We get 2 cells, and then we start over. So we're focusing in the timeline of a cell's life, we're focusing just on this portion right here. So how exactly does that work? Well, in the last lecture, there was a quick video, not really quick, but there was a video that talked about the historical context of the discovery of the helical model of DNA. So Watson and Crick's Nobel Prize winning research. This is a side branch from that same video series that really just focuses on the mechanism by which, the three-dimensional visualization by which DNA splits and replicates itself. And it really helps understand the idea of semi-conservatism. In semi-conservatism, every time you create a new strand of DNA, half of it is from the parent DNA and half of it is new. So it's conservative in that it's conserving the original material and reusing it. So let's take a look at this for just a second. Oops, I have the video off, hold on, or I have the sound off. Okay.
The structure of DNA that Watson and Crick discovered suggests how genetic information is passed on. Before a cell divides, the double helix unwinds and the two strands of the DNA molecule in the nucleus separate. Each strand is then used as a template for the construction of new DNA molecules. The replication of DNA is simple in theory, but much more complicated in reality. The precise details have only recently been worked out. Take a look at the animation called replication mechanism to see what actually happens at the molecular level.
Okay. I paused right here because this is really what we're interested in. So our original strand is up here. It is unwound. Now, there are specific molecules that we're going to talk about, enzymes, that help that process, but you unwind the helix and you break the hydrogen bonds along the center. And what that causes is the entire strand is split in half. So you have one strand that goes left and one strand that goes right. Now, there are consequences when we talk about the strands being anti-parallel, and we'll talk about that in a few minutes. But this is a really easy conceptual model to understand semi-conservatism. You have the original strand, you split it down the middle. One half of the old strand becomes half of the new strand. The other half of the old strand becomes half of a new strand. And you build the new strands along the old strands. Now, because of those Chagraph rules with A's and G's and T's and C's, each strand acts as a template to build the new one, and you get two exact copies. So this is kind of a summary slide that talks about that process. You have your original parent strands. Now you'll see the 5' and the 3' ends, and that has to do with the direction that the strand is going, the way that those ribose sugars or deoxyribose sugars are pointed. They point towards the 5' end. Now, these numbers become important when you're talking about DNA replication because DNA polymerase, the enzyme that actually adds the base pairs on, it can only work in one direction. So those directions matter. It can only drive on the right side of the street. So here we have our original parent strand. We have it split. The original strand becomes one half of one daughter strand. Here we have half of the parent strand becoming a part of another new daughter strand, and the new DNA is synthesized in the middle. That is semi-conservative, meaning half conservative. Full conservative would be you have one whole strand that stays the same and one whole brand new strand. That would be fully conservative. Now, this only works because of Chagraph's rules for A's, G's, T's, and C's. On our original strand, we have, let me change my color. Right, so we have A's have to be with T's and G's have to be with C's. This has 3 base pairs, so it's got to be a G and a C, right? So if this is a C, that means that has to be a G, right? So when we split it, here's our C. Well, how does that polymerase know what goes here? Well, it can only be a G. Same thing here. So here we have our G that's on the parent, and when DNA polymerase comes by, it has to add a C because that's the only thing that can go there. So when we say that the result is two identical strands because of complementary base pairing, it's this rule, it's Chagraph's rules that ensure you get two exact copies of the parent. Without those rules, you wouldn't have exact copies. You'd have lots of changes. All right. On a scale of 1 to 5, 1 being like, I don't know, I'm not even sitting in front of my computer. 5 being like, yeah, I totally got this. Can we keep going? How do you feel about what we're doing so far? Amy says 5. You can put it in the chat or you can put it in. There you go. You can give me a symbol. Grant says he's good. And let's see. Somebody's in the chat window. Good. James says he's good. Okay. All right. We're going to keep going. So here we have 3 different models that were originally entertained when we started thinking about how DNA copies itself. Now, I already told you that semi-conservative is the answer, right? But we also considered 2 other models called conservative and dispersive. So in a conservative model, after the first DNA replication, you'd have one that is essentially just all parent material and one that's brand new. And the same thing would happen over again. You'd have the original parent material and then the rest that are brand new. Dispersive, it's a mix. You can kind of think of this as like crossing over, where you have like bits and pieces in both that are new and old. We know now that conservative is the model where you have half and half with each new generation. And it's really the Messelson and Stahl experiment that kind of nailed this and proved it. Now, this experiment is kind of stunningly simple. You have DNA, which is composed of carbon in the ribose. You've got hydrogen that's also in the ribose. You've got phosphorus in the phosphate group, and you've got nitrogen in the base pair. So if I were to put that together, it would look like your phosphate, right? Your nucleate, your ribose, deoxyribose, and your base pair. So the nitrogen is in this part. When cells create new DNA, they obtain that nitrogen from the food that you eat. Right? So the DNA in food that you eat, your body breaks it down into nucleotides and then uses those nucleotides to build your DNA. So when they say you are what you eat, you are actually what you eat. So the nucleotides from like the carrot that you ate for lunch are being incorporated into your own DNA as you speak, or as I speak. So Messelson and Stahl knew this, so they used bacteria. So they cultured bacteria in a broth in which the nitrogen in that broth was heavy. So it has an added neutron. So it's heavier than it's supposed to be, right? That's the only difference. It's a little bit heavier. After a couple generations, when all the bacteria had DNA that was pretty much loaded with nitrogen 15, they took it out and they put it in broth with a lighter version of nitrogen. Now, bacteria duplicate over varying timescales. Some, like E. coli, duplicates every like 20 minutes. Other types of bacteria, they duplicate once every like 12 or 13 days. I'm not sure which bacteria they use. They probably use E. coli. So they let it sit in the broth, the light broth, for about 20 minutes and let the cells divide approximately 1 time, right? So they have 1 new generation. And they took it out and they centrifuged the DNA. Now, under a conservative mechanism, you would expect to see in their centrifuge tubes. So let's see. Centrifuge tube. In a conservative mechanism, you would expect to see all the old DNA would be towards the bottom of the test tube because it's heavy. Right? All the new DNA would be higher up because it's lighter. But that's not what they saw. Instead, they saw in their test tube, centrifuge tube, and centrifuge means like they spun it around, right? Have you guys ever been on that ride at the fair where like you put your hands against the wall and then the floor drops out and it spins around and you have to like stick to the wall using centripetal force? Same thing, right? And all the heavy stuff goes to the bottom and the light stuff goes to the top. So when they centrifuged, I shouldn't do it there, I should do it down here. When they centrifuged this stuff, what they got was one line in the middle. Because each strand of DNA was half light and half heavy. So it was neither light nor heavy, it was right in the middle. And they continued that process over and over again, and they came up with some interesting ratios. I'm going to stop for a second. Somebody's got to have a question about this because this is like not straightforward. So tell me what questions you have. You can put it in the chat if you don't want to speak.
Can you tell me why they only had a singular strand?
To start with?
No, like when they opened the centrifuge.
Oh, they didn't. They had a whole bunch. But when they centrifuged it out, it separated. So they took a colony of bacteria and did this. So they took a colony of, here we go back. So they took an entire colony of bacteria. They cultured it and grew it in the N15. So they had like, a few million bacteria in N15. They took it out, all of it, and they dumped it at N14. And they let it sit for 20 minutes because they divide every 20 minutes. And then after 20 minutes, they didn't pull all of it out. They pulled a sample of it out. And they centrifuged the sample. So you get DNA by, it's actually really simple. You take a little bit of shampoo and you put shampoo in with the bacteria, and the shampoo dissolves the lipids of the cell membranes and the lipids of the nuclear membrane, and it spills all the DNA out. And then you centrifuge the crap out of it, and everything separates out by density. And you know exactly where to pull the DNA from. So what they found is, for the DNA, they got rid of a bunch of other stuff too. They used lysosomes and all that other good stuff. So that all they had left was DNA. DNA is kind of like snot. When you isolate it by itself, it's really gross and sticky. So after the 20 minutes, they isolated the DNA, centrifuged it, and this is what they got. So are you saying, why did they only get this? Why don't they have any of this left over?
Yeah.
Because so this was the parent generation, right? After 20 minutes, what does the parent generation do?
It multiplies again.
Right. In order to do that, it has to copy its DNA. So all of the parents go through that process and give you this.
Oh, okay.
Yeah, there's no more parents left anymore.
Okay.
So then when they do it again, when they put these guys, where they some of those bacteria are still in the light broth, they do it again and they come up with this. That's a good question. Why do they only have this? Because all these guys are gone. So this is another way to look at it. So here's the original parent material. They duplicate in the lighter broth. So the only nitrogen available to them is the lighter nitrogen. And for this entire generation, you have DNA that's basically hybrid, half heavy and half light. So the band moves up a little bit. Now, these guys, right, the ones that are still in the light broth, they're going to duplicate again. And because of semi-conservatism, half, so the pink strand's going to go here, and new light DNA is going to form, and our parent strand goes here, right? And it's got N14, and then same thing here. We have our pink strand, and you have got new strand, and then here's our blue strand, and you have more blue strand. So then you can see two stripes. You have your original two strands are still there, right? That's why it's conservative. Our original two strands are still there, but everything else is light. So that's semi-conservatism. Okay, on a scale of 1 to 5, 1 being like, I am not paying attention. 3 being like, I sort of kind of get it, but I'm a little unsure. 5 being like, yep, makes total sense. I could totally nail a question on this. How do you feel? I see 3s and 4s. I see a 4. I see a 4. Okay. Alrighty. So here's another way to look at it. You can actually calculate the weights. So here we have 100% heavy, right? We have 100% that's medium. And now you start to see a split from there. 50% is heavy and 50% is split. And every generation thereafter, you take a fraction of half and add it to the top. So the next one would be 6% light, right? And 94%, or I'm sorry, 6% heavy and 94% light, because you keep going. Just another graphic that shows you the exact same thing. Just another way to think about it. Because of this half and half ratio, where half of it keeps moving up, that's what really nailed the semi-conservative, meaning half of each strand is being replicated each time. You're conserving the old half, and new half is being replicated each time. Okay, so now we're going to move on to like the nitty-gritty parts and pieces of how the DNA actually replicates itself. And there are a couple of different enzymes that are included. Understanding 2 includes helicase, which unwinds the double helix and separates the two strands by breaking down the hydrogen bonds. And understanding number 3 deals with DNA polymerase, which is the molecule that's going to add nucleotides to the new strand. So here we have a strand of DNA, and we said a few minutes ago that, remember, DNA is anti-parallel. It's got sides of the street where you only travel in one direction on each side of the street. The ribose points towards the 5' end. So if that's 5' on that end, that means it's got to be 3' on this end. Now, the other half of the strand has to be going in the opposite direction. So that's 3' and 5'. And this becomes important because certain molecules can only work in certain directions. They can only work from 5 to 3. DNA helicase is not one of those. DNA helicase doesn't care what the directions are. Now, first off, helicase tells you what it does in the word. So ASE, remember, anything that ends with ASE is an enzyme. And enzymes either build things or break things. That's their job. So helicase, right? It's going to be breaking the helix. DNA helicase, it's breaking the helix of DNA. DNA helicase breaks the bonds through the center, breaks those hydrogen bonds that hold those base pairs together. So I have scissors as my helicase. It's going to come through and cut those hydrogen bonds. And separate our strands. Now the next step deals with polymerase. DNA polymerase. Again, we have an enzyme, ASE, right? You know it's an enzyme because it's got that ASE. Polymer. Okay, we have an enzyme that is building polymers. What kind of polymers? It's building DNA. So DNA polymerase is going to be building DNA molecules. It is the thing that brings the nucleotides to the strand to build the daughter strand, right? Now you notice this is happening in a certain order. This strand is anti-parallel, so that means that's the 5' and that's the 3'. So you'll hear questions, or you'll see questions that'll ask you, what direction does DNA form in? Now you have to be specific with reference to the strand. So DNA polymerase works from 5 to 3 on the template strand. Let me write that on here. Oops, I can spell. It's hard to write sideways on a surface. That's supposed to say daughter. There we go. So DNA polymerase works from 5 to 3 on the template strand, which means if you're talking about the daughter strand, it's what direction? I'll give you a big hint. It's the opposite.
5 to 3.
Nope, that's the same.
Oh, wait, no. Did you say 5 to 3? Did you just say 5 to 3?
Yeah.
Oh, no, sorry. I meant to say 3 to 5.
Yep. There we go. Right? So DNA polymerase works on the template strand in the 5 to 3 direction. Now, if you're talking about with respect to the daughter strand, it's the 3 to 5 direction. Now, that becomes important because on this side, you have the opposite. You have the opposite direction. And DNA polymerase can only work in one direction. It can't read from left to right and right to left. So that causes a problem during the replication process that you'll see in a few minutes. But let's add them anyways. Let's pretend that's not a problem right now. The last step is DNA ligase. And DNA ligase is going to reconnect those strands by reforming those hydrogen bonds between the base pairs. Maybe we should put that. There. This is why I type everything, because my handwriting when I'm drawing on the screen is rather horrible. Which is weird, because if you look at my notes are pristine. Different. Okay, how do you feel about this? Steps 1, 2, and 3. Okay, good. Because I have seen test questions that ask you to explain the process of DNA replication, and as long as you can describe these 3 steps in 3 steps with the relevant information, 5 to 3, 3 to 5, complementary base pairs, then you should be okay. So now we have an animation that's actually going to show you, you may remember this from freshman year if you had me freshman year. We have the incoming strand of DNA. We have our helicase, this blue molecule right here that is going to work to split the helix and separate the strands. And then we have 2 strands. We have a leading strand and a lagging strand. The leading strand, DNA polymerase is going to add the base pairs on nice and smooth. Actually, that's right here. And then, so this is the DNA polymerase that's adding the base pairs. And then this green molecule here is the ligase that's reforming those hydrogen bonds. Okay, so we've got our helicase here. We've got polymerase, DNA polymerase, that's adding those nucleotides. And we have the ligase right here that's reforming those hydrogen bonds. Now, I use this video because I like it. It does a good job of showing you the problem with the lagging strand because DNA polymerase can't work backwards. So we'll watch that happen.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems.
Just FYI, that's like as fast as a jet engine. And this is happening in pretty much all of your cells, or a large majority of your cells. It's happening continuously in your body, right? Not all your cells at the same time, but it's happening. Any cell that's going through S phase, this is happening in.
The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Okay, I'm going to back this up for a second and freeze it.
Intricate as this mechanism appears, on back for replication.
Okay. All right. So here we have our parent strand. This is coming in. Here's our helicase that is splitting. Now our leading strand goes off this way. And it's not highlighted right now, but that's okay. The leading strand is really easy. Here's your DNA polymerase. Here's your ligase. And because it's the leading strand, that polymerase can work in its preferred orientation, and it just keeps going. You can think of it as like if you're typing in a document, the document, you could just keep typing forever, and your sentence will just keep going from left to right forever. And you never have to hit enter and come back. You can just keep going because you write in that direction. The lagging strand is different. The lagging strand, you can write for a little bit, and then you have to hit enter and go back and start typing again, and then you hit the end of the page, and you have to go back. You hit enter to go back to the beginning, and then you write to the end of the page. That's exactly how DNA polymerase works on the lagging strand. So
Kind of like a printer or a typewriter.
It's exactly like a typewriter, right? So you type on a typewriter, and then you have to push the register back and start typing again. So here we have, you can see the lagging strand, the helicase splits it, and it lets a big loop out, and it loops back. And then you can see here is a DNA polymerase, and it's going to work backwards for a little bit. And if you look back here, you can see there's another strand that has been replicated, and there's a big chunk in between that's missing. And if I hit play, you'll see it'll work all the way up to that point, and then it stops, right? So it's going to let a bunch go, and then it writes until it connects to the last spot where it started. So it effectively has to work backwards. Now that's a little complicated, but it was part of the freshman year stuff. We did the 5' 3' thing. And I think as long as you understand that DNA polymerase on the lagging strand has to work backwards, and those chunks that are formed are called Okazaki fragments, then you should be okay. If you want more information about that particular process, but that was good, Grant. It's exactly like a typewriter. Like that's the analogy that I use when I think about it. So let's see. Let me bring this back over here. There's a crash course video on the Canvas page, and it's kind of long. It's like 12 minutes. You don't have to watch the whole thing. If you do want to watch the whole thing, it's a good refresher from the last lecture. It talks about everything last lecture and this lecture. So it's the two combined in like 15 minutes. If you don't want to watch the whole thing, if you just go to like 8 minutes in, it gives you a better visualization, or not better, just another visualization of how that process works, where you have one strand that's formed continuously, where you just keep typing and it just keeps going. That's the leading strand. Whereas the lagging strand, you have to type to the end and then go back and type to the end and then go back. So do you want to watch that now, or do you guys want to do that on your own? Because I want to make sure I get you started on the activity before the end of the day.
For the recording's sake, I feel like we can skip it, so it's not like a big chunk of,
Okay.
video.
That's perfect. That's perfectly fine. So if you're at your leisure, go back and watch that if you have questions about that particular process. So then at the bottom, once you take your notes for U1, 2, 3, and S2, go ahead and submit those. And then if you hit next, the next page. This is a quick little activity that deals with the Mendelssohn-Stahl experiment, and that's the experiment that showed semi-conservatism that we talked about. Now, you have a couple of different choices here. You can download the PDF and print it out, or you can download and type on it if you have Adobe, if you have access to tools in Adobe. Or you can draw your own answers on a sheet of paper and take photos and upload it. Or if you want to create it on a Word document, if you want to like screen clip and paste pieces and then put it on a Word document, I'm fine with that. I don't care how you do it. Be as creative as you want. What you do need to do is you can work independently. So if you want to sign off after this and go do it by yourself, you are welcome to do that. If you want to stay on and work together, that's fine too. There's some reading that goes through the differences between conservative, semi-conservative, and dispersive replication. It talks a little bit about the Mendelssohn-Stahl experiment, which you'll also see called the pulse chase experiment, same thing. And it has you use colors to kind of demonstrate the process of semi-conservative replication. So you're going to need, if you're using pen and paper, you're going to need a couple of different colored pencils or markers. And there's some questions. But all you're doing is following the instructions and answering the questions, and you have the choice to work together or not. So that's it. I wanted to make sure I got that explained for you. You can upload your document to make sure you get credit. If you would like to work independently, you are welcome to sign off now. If you would like to stay on and work together, you're welcome to do that too. Okay? All right. And of course, I'm here to answer any questions.
Bye.
Bye.